found out a minute ago or so that I was chairing this session, so excuse me for not giving a very long introduction to the next speaker. This is John Aston, who works at Cambridge at the Statistical Laboratory, which is you right here. And he'll be talking about inverting entomological growth curves using functional data analysis.
Well, thanks very much for the invitation to be here. I think in the spirit of all the disclaimers that were given in the last session, I probably am a mathematician, and which means that on the other hand, I know nothing whatsoever about forensics, so please be gentle if you think I've made a howler, because I probably have. So yes, as Maja said, I work over in the maths department just over there, and this is very much joint work. This is joint work with Davide Pigoli, who is a postdoc here in Cambridge, who's done most of the actual work on this. Anjali Mazumder, who is at CMU and who some of you will know, and she's the one who really sort of formed the link between the statistics and the forensics. Frederic, Ferraty, who's a statistician in Toulouse, and then Martin Hall, who's a forensic entomologist at the Natural History Museum and does most of the UK's forensic entomology work, particularly the kind of things that I'm going to be talking about. So, it's a slightly sort of long and involved title, but it's really going to be talking today about forensic entomology, or bugs. And while I like to, when I give a talk, show the data as the first kind of thing, I think probably as we're going to have lunch in a few minutes, you probably don't actually want to see the data in this case. These are pretty unpleasant scenes that Martin gets called to go to. And what Martin's job is to do is to try and determine at least the time post-mortem interval for cases where bodies have been around for a while, and therefore sort of traditional pathological evidence is not particularly reliable. So what kind of data do we actually get to look at? Well, when Martin goes to a crime scene, he collects a standard set of evidence. So he collects the temperature data at the crime scene after the discovery of the body. So he of course doesn't know what body, the temperature that the body has been exposed to prior to where it is discovered, but assuming that the body has been left relatively stable position, he wants to understand what the temperature process is post discovery in order to be able to try and infer the temperature process prior to that. And so what he then uses is the temperature data from nearby weather stations, which he also collects, and then hopefully, so one part of the talk, only a very small part, but one part of the talk will be about building models that allow you to try and predict the temperature back from the temperature data that we have at nearby weather stations. And that's going to be incredibly important because of the fact that we're going to be interested in looking at particularly blowfly growth. And blowfly growth is incredibly temperature sensitive. And so we need to be able to build models that allow us to understand how these blowflies grow in order to estimate time of growth, so that we can look at the observed data to try and get an estimate of the post-mortem interval, or at least time to colonization, which is what we can officially get out of the data. And in order to do that, we need to understand how flies grow at different temperatures and what kind of growth curves they might be might be arising in particular temperature situations. And then finally, of course, we get the entomological data that's collected at the crime scene. These tend to be maggot lengths, maggot stages, and looking to see whether there are other entomological evidence like pupa that have been discarded, so assuming that there's been a whole life cycle going through of blowfly, and using that to try and date when the body was placed at the scene of the crime. So most of the work we're going to look at is for blowflies, although I'll talk about multi-species stuff very briefly at the end. The idea is that for a particular constant temperature, blowflies behave very regularly. So after eggs are laid, after about a day, they hatch and become maggots, and then they have different stages in stars of development, and these stages are effectively where additional holes grow in the larvae, and these happen at fairly regular times, and it's all to do with thermal input. The blowflies need a certain amount of thermal input to go from eggs all the way through to pupation and then finally to an adult fly. And so it's quite easy to do this once you know what the constant temperature is, and in actual fact sort of very relatively simple models allow you to take a constant temperature blowfly eggs and work out at what stage they're in by looking at their length or looking at their weight or looking at their development stage. And so that's one way to then look to do time of death or post-mortem interval by estimating the time from colonization. Colonization happens very quickly. Martin's run a large number of experiments of bodies in particular different situations, and it's very, very quick that colonization, even sitting outside his window 8 floors up, put something outside, within a couple of hours it will be colonized. So yeah, indoor bodies, so it's the same principle. He's even done situations where he's locked well, not bodies, locked pigs, carcasses inside locked suitcases that you would think would be airtight, and 24 hours later there will be insects inside. These are good at getting into places. So it's quite a useful technique, and so what we want to be able to do is want to be able to replicate this, but in situations where, of course, we don't get to see constant temperatures and the conditions are much more complex. So we have an awful lot of raw experimental data. This is supposed to show that it's kind of nasty data, but in many ways you can kind of see what's going on. What's happening here, we've got the time in hours, and here we've got various different colors representing various different temperatures, and there are some repeated colors, I apologize for that. But what you can see is, for example, if we look at 27 degrees here, which is this set of blue temperatures here, we see that within under 200 hours, the flies have gone from being eggs all the way through to pupation. They're being pupated out and started to become the process of metamorphosis into an adult fly, whereas some of the ones down here at, say, 2 degrees have hardly grown anything even after sort of 800 hours, and they actually just stop development. Development doesn't continue, and even the ones at maybe 4.5 degrees or maybe 7.5 degrees, they do reach pupation, but they take 6, 8 times as long as the flies that were at a much higher temperature. And so what we would like to be able to do is take these different experimental data. Each dot here represents a particular fly that went through the process at a particular constant temperature, and use that to try and understand what's going on in different situations. So probably more interesting or more easy to look at is some averages. So here we've got on two sides. So the length of the flies here is in the black, and this is the black curve. The in star, which is the level of development, going from 0 to 6, 6 being full pupation and adulthood. We see that in some situations, up in the high temperatures, we have very, very fast growth and very fast development, and they reach adulthood very, very quickly, whereas in very low temperatures, they may never reach adulthood, or if they do, they take a very long time to get there, much longer. Yeah.
If the fly starts out cold and then it gets warm,
good question.
will it speed up?
Good question. So that's exactly what we want to know. So that's because, of course, what will happen is, for example, if you find a temperature body outside during the day-night fluctuations, you're going to have big changes in temperature. So that's exactly the problem, in the sense that Martin doesn't get to see constant temperature. What he would like to know is build profiles of how these things happen when you have very large fluctuations in temperature.
Can you just explain the red and the black on these
Sure,
diagrams?
of course. So the black is simply the length. So it's this side. Look at this side. It's just the length of the larvae as they grow. The black is the black. The red is the instar stage, the developmental stage that each fly, and it's on this side here. So the red axis is this one. The black axis is this one. Yeah. So, and exactly to answer your question, this is the kind of temperature profiles that we see at particular locations. So this is a particular crime scene that I'll come on to later, and what you see is you have a very, very variable temperature going on. Exactly, day-night is a very good description of that, but there are other things that sort of change it depending on the time of the year, etc. So this is the temperature profile that you get to see. This is the temperature profile of the weather station, with the red bit here being simply corresponding to the same time frame as the blue up here. And what we'd like to be able to do is try and fill in the blank here so that we have the temperature profile for the crime scene pre-body discovery, so that we can then use that to try and understand the process that these flies will go through in order to be able to accurately generate an individual growth curve for a set of flies. So we had the nice growth curves there, or at least what looked like, if you join them up, could well be growth curves for a fly at, say, 12.5 degrees. It's got a nice growth curve here. It does have a drop at the end, which comes from the fact that once the larvae have eaten their fill, they wander off to find somewhere safe to pupate, and so therefore they do actually decrease in body mass. So it's not just an increase in function, it's not just an increase in growth. They do is actually a drop at the end. But what we can see is there are these sort of growth curves at individual temperatures that seem to change speed. depending on what the temperature is. And what we'd like to be able to do is look to see, use these weather station predictions to generate a temperature prediction here that would allow us to build an individual growth curve for a fly that or an egg and larva that was subjected to that particular temperature process. So I'm going to talk about sort of three different bits that need to go into doing this. So the first bit is we need to build a model for temperature, and I'm just going to talk about a couple of very simple models, and you can imagine there's all sorts of more complicated ways you can do this, but it's not really going to be the focus of what we're going to talk about. Then the second one is much more to address the exact point that you raised, the fact that we really need to build temperature-specific growth curves for non-constant temperatures using the experimental data we have in the lab from those temperatures, those constant temperature profiles. And then third one is how we then apply that to the crime scene evidence that we had collect, the observed lengths. So we just get to see a single point in time, look to see what's the length of those maggots at that time, and then look to see where could they have come from given a particular set of possible profiles, because we don't know when the start happened. So they could have been subjected to different temperature profiles depending on where you decide the start or the end of the crime scene happened. And then we look at some things that I've obviously missed out that I think we need to talk about. And I think this is somewhat preliminary work, so I would be very interested in people's ideas and points that we think we need to consider that we haven't. So, very simple for temperature modelling. Well, the current method that's used is simply a standard linear regression. You take the weather station data, you regress it on the known logged temperature data. So they tend to place a temperature logger at the crime scene for about 5 days to get an accurate comparison, and then they do regression on the 5 days worth of data between the weather stations and the 5 days recorded at from the temperature logger, and then apply those regression coefficients backwards to get a backwards prediction of the crime scene temperature. And you can see that you just get to sort of fill in the blanks as you would with the standard linear regression model. So that's fairly straightforward. We sort of looked what we could do. Could we do any better? Well, you can do a little bit better if you take a few more add a few more bells and whistles. You take into account that things seem to behave differently day and night. Maybe use some kind of non-parametric regression to sort of capture the ups and downs a bit better, not make it go quite so high at some times and quite so low at others. So here we see the black curve is the data. The black light curve here is of just using an observed temperature station, so finding the nearest matching temperature weather station and just filling it in using that. The red curve here is just using the linear regression, and the green curve here is using a non-parametric regression based on spline regression, and you get some kind of nice, slightly less sharp peaks, but it seems to sort of fit a fairly standard temperature profile. And I think one of the things that we should incorporate, which I'll talk about towards the end, is how you might incorporate the errors in estimating these temperatures, because they will flow through to the estimates, the error estimates on estimating your temperature curves, which will then flow through to the estimate of your post-mortem interval. So we have this relatively simple model that we can get some kind of temperature prediction out, but of course we don't know where we should start our temperatures from, because we don't know the post-mortem interval, so we've got to sort of somehow say, well, where are we going to start our temperature curves from in order to build the possible growth profile of the flies, so that we can then look to see whether we can compare them against the observed flies.
Can I just ask, how long do the flies last? I mean, if you find a body a year later, presumably it's all finished.
Yeah, well, so that's a good question. So, where a particular fly, I mean, depends what temperature it's been subjected to, but it's the same. It's in a reasonable range of temperatures. Yes, the flies would have come and gone, and then they. However, there are other entomological evidence, beetles, for example, that go for much longer. So you can actually use the same approach for multiple different species to look at different time frames. So there's still entomological evidence available even after a year or more at this point, but not necessarily flies. So I wasn't quite sure how much people here would know about functional data analysis, so I thought I'd give a very quick introduction to functional data analysis, and apologies if people know about this already. But functional data analysis sort of comes out of the sort of school of multivariate statistics. We basically, from almost everybody here will be familiar with multivariate statistics, where you have multiple measurements that are highly correlated with each other in many cases. Here's the sort of very traditional Fisher iris data back to 1936, where he had 3 different types of iris, and you can see that they are well separated by using various different combinations of the 4 measurements of sepal length, sepal width, petal length, and petal width. Now, that is a nice sort of example of where using a vector of observations, in this case 4 vector of observations, allows you to do something with your data. Functional data analysis is somewhat just basically can be the sort of most simply thought of an extension of this, where instead of having 4 or any kind of number dimension, you assume there are an infinite number of possible multivariate observations that you could have, if you were able to go as finely as possible, able to look at. So one particular easy and sort of early form of functional data to kind of visualize is this, which is the observed records of angular rotation of the hip. So if I do one gait motion as I walk through the stage and look at the angle that my hip is making with my leg, then this is the angle that is happening across the start to finish of one gait cycle. Now, there is no reason why I need to, if I wanted, I could take a measurement of this every 10th of my gait cycle, I could take a measurement every 100th of my gait cycle, I could take it every 1000th, I could take it any discretization I like, and what I would hope is that there would still be a measurement that would give me some kind of smooth curve underneath. And the whole idea about functional data analysis is to sort of assume that there is some kind of underlying smooth curve underlying this whole system, and that the discretization doesn't matter. The other thing that functional data analysis has about it, which is quite important for this, for the life cycle data that we'll look at for the flies, is that in actual fact we also aren't particularly worried about whether time moves around it or not. In that the function can be stretched, it can be compressed, and we'll still believe that in underlying it has the same kind of properties. And we'll look and see that that actually is very important in here, because that's effectively what the role of temperature does. It just shrinks or contracts what's underlying biological time.
Can I just ask,
Yeah.
if you could measure, say, calorific input, would it be a fixed picture, irrespective of?
Very close to being a fixed picture. In actual fact, that's exactly the model that they use at the moment, so let's show that in a second. Yeah. So,
could you look into Arrhenius equations?
sorry?
Into the Arrhenius equation, which is the speed of a reaction over the temperature, which is a way to stretch and to...
You'll see it's very similar to that in a second. So yeah, very similar. So modern functional data analysis goes beyond just curves and surfaces, and they bring into consideration all sorts of different things, whether it's multi-dimensional data, so you've got images, for example, sounds being another way of looking at this, data that lies in particular constrained spaces, monotonic functions, which is good for part of our growth curve data, but in actual fact misses out this sort of drop at the end, which I was talking about earlier. But modern functional data analysis allows you to look at all these different types. So in actual fact, it sort of could be useful in other situations as well. I think there are other situations where you have evidence that could be pictures or particularly images where this would naturally fit in the same kind of framework. But going back to our bugs, what is the model that is currently used? And this goes exactly to Phil's point, that basically they use an accumulated 3-hour model. So basically they say we know how long it takes for a fly to go from being an egg through to pupation, and that takes effectively a fixed amount of thermal input. And so therefore you can actually look to see what that fixed amount of thermal input is in terms of some kind of amount of time and some kind of temperature that is available for the fly to be seen. Now what they currently do is take the profile over the time that you're interested in, take an average and use that single number as the average, not worrying about the fact that it has oscillations in it. And what they look at is they take the average. They know that below about 2 degrees, flies don't grow. They kind of go into stasis. They don't. They no longer grow. They no longer even evolve at a small amount of time. So therefore, there is some kind of bottom limit. But above that, this accumulated degree hours says I need so many degree hours to reach pupation or reach adulthood, and then I can use that to backtrack. So what you do is when you find your evidence at the crime scene, you collect some samples, you take them back to the lab, you put them at a constant temperature, you feed them, put them in sort of, usually about 70 degrees, and then you watch to see how long it takes for them to develop into adult flies. What you then do is take off the amount of thermal input, the amount of accumulated degree hours that they've had in the lab, and you then divide it through by the temperature so that gives you, given you know the total amount of degree hours that is needed to turn a fly from egg through to adult. You take off the amount from the lab, you divide it through by the average temperature at the crime scene, and that gives you an estimate of the post-mortem time interval. So it gives you a way of taking the data, but it requires a couple of things. It requires that you have a pretty good idea of the accumulated degree hours. Well, that's fine. That's fairly well studied biologically, and it's pretty good. But the other thing that you require is the fact that the temperature in this model is constant, and it's not necessarily a good model for doing that. The last thing, of course, is you have to wait. You have to wait for the flies to sit there and turn into adults from the specimens that you collected, which may take a while, and therefore you may have lost time in being able to decide what the post-mortem time interval, which could have effects on whether, if the police are holding a suspect, for example. So we wanted to try this a different way, taking into account the full kind of temperature growth curve that you could see in the data. So this is a bit notation heavy, so I apologise, but basically what you say is we've got a whole bunch of temperatures collected at particular times, and these times correspond to possible times of discovery. We don't know what the time of discovery is, and we're interested in defining the time of discovery, and then there's this X tilde process, which is basically the temperatures that this fly has been subjected to up to time K. So it's what the different temperatures over some discretized intervals, so we're going to use hours, what the hourly temperatures that the fly has been exposed to up to that point, and then we're going to let L X tilde K T be the length of the maggot that we observe at time Tk, having been subjected to the previous temperature process that this maggot has been subjected to from the beginning of time. Well, of course, we don't know what the beginning of time is, and then let n star be that unknown time. So when we take our data, we observe it at time n star, which we don't know. It's been subjected to some unknown process of temperature, which we don't know. And what we would like to do is try and infer this n star, because once we know what n star is, we can work backwards and we can figure out exactly what all the different, growth profile would look like for that particular maggot, and then what particular lengths we would expect to see from those maggots which we collected. And typically, Martin gets somewhere between 8 to 15 maggot observations that he gets to look at. They're typically the biggest ones he finds on the body, because as you can imagine, things don't sort of stop once the first flies arrive. Once the first flies arrive, the next set of flies arrive, and the next set, and so there's constantly maggots being formed on the body. But if you believe that if you don't find any evidence of pupation already, then you believe that the underlying largest maggots are probably from the first flies that colonize the body. So that's the sort of way. So he looks for the sort of 10 biggest ones and says, what are they? And then uses that as the idea of the first set of maggots that have arrived. So we have a temperature logger that logs the temperature from time n star onwards for up to some number of observations, maybe 5 days, which is what the normal temperature logger does. And then we have a bunch of experimental temperatures that we get to see the data collected in the lab. This is data that's from start to finish. This is experiments run on flies for a known temperature from the beginning of egg laying all the way through to adulthood. And we get to see what happens, what the length curves, what the growth curves are for those individual set of temperatures. So in our case, it was 2.5, 4 degrees all the way up to 27 degrees. So we have individual growth curve profiles, and we want to somehow build a model that takes into account the fact that we want to know that for varying temperature, how we could build an individualized profile for a particular curve. So the way we're going to do that is basically looking at speed. And it seems to be a fairly biologically reasonable model that the growth of a fly at a particular time purely depends on how big it is at that time and the current temperature that it is exposed to. Well, you can go backwards, and this just says, well, I'm going to take a few temperatures into account. So this could be taking a capital B number of temperatures in the past into account. But if you want to think about this as WB just being 1, just being instantaneous, this just says that the rate of change, the speed that my fly will grow at this particular time is purely dependent on the temperature at that time, xk, and also the length it is at that particular time. This is just inverting the length profile and saying at that particular length at this particular temperature, how fast does it grow? And we can get that from our experimental evidence for the evidential, for the particular temperatures that we've actually done in the lab. So what we want to be able to do is say, if I happen to have a fly that I'm currently looking at that's 5 millimeters long and is currently subject to 27 degrees, I go to the experimental profile of a 27 degree fly, look to see how fast it grows at that rate, and I use that in my model currently to see how far it's going to move, how fast it's going to grow in the next hour. And then you can think about it, well, that just makes it very easy if I make an approximation about time over sort of hours, about how to build a single temperature dependent growth profile for any fly that I observe for any particular length of time. I start with it at some particular temperature, and then I let it, for that particular temperature, then each hour in between, I just move the growth profile depending on the current temperature at that time that I've seen, and then just do that point by point by point up to time k, and that gives me the accumulated length that I would expect to see for the growth of the curve at any particular time. And then all we want to do is we want to take the minimum over all my observed lengths and the lengths that come from any particular profile I look at, find the minimum value for n star, and then that gives me my post-mortem time interval. So problem is that we only know a small number of constant temperatures, so therefore we have a 4-stage procedure for estimation. So the first stage is to somehow fill in the gaps, because they only get these temperatures, these growth profiles only going to get recorded every 24 hours when someone comes and measures each individual set of maggots. Well, that's fine, we can just do some kind of non-parametric regression to fill in the gaps, so therefore we get these smooth temperature profiles for each individual temperature. But now if you look at those profiles kind of look similar, apart from the fact they're going down slightly so that the ones with a smaller temperature tend to not get quite so big. Apart from that, they all have this kind of similar shape. They rise steeply and then they drop off. And I can imagine that I could just sort of move time around, and I could make this curve look very much like this curve simply by shrinking this dimension on my time axis. By squashing the axis together, I could turn that curve into one of those curves. So that's exactly what we do. We say, can I make all those curves line up, and can I change time? And these curves here simply tell me how to change time. It says for curves of higher temperature, run time faster. For curves of lower temperature, run time more slowly. And if I do that, then these curves can all line up, and then I can get pretty good estimates of what's going on in these curves for any particular temperature, and I can get pretty good estimates in here of how to change time for any particular temperature simply by averaging between them. So I average between the curves for particular temperatures. So how do I find out what curve at let's say 13 degrees is? Well, I take the temperature at 12.5 degrees curve, and I take the curve at 15 degrees. I take a linear combination of the two observed temperatures, but I also take a linear combination of the speeds of the shrinking or stretching process that I need to do to time, and that's what I get here. So each one of these curves represents a particular temperature in between my observed experimental temperatures, and these show the growth curves normalized by time for each of my experimental temperatures. And then if I want to get back a particular curve at a particular observed curve, I simply invert the temperature and stretching process. So this now gives me a set of profile temperature curves that renormalize, that turn time back from this squashed or stretched time into real time. That's what this set of curves do here. And then I apply it to my growth curves, my actual growth curves, and that gives me this set of growth curves across any temperature that I particularly would like. So what does that allow me to do? Well, it allows me for any particular time in the past, so for any particular length, to say how can I run backwards. So if I combine my speed curve, which depends purely on temperature, and I combine this set of curves here, which says for any particular temperature now, I can give you what the speed is at any particular point. At this point, I can calculate the local derivative and use that to calculate speed for this particular given temperature. I can then plug it back into my model that will give me a growth curve for the entire, for that particular time, for that particular maggot. I can then, for particular lengths, look to see which growth curve profiles seem to fit that right. So this is for, this one here, for example, is for one that seems to get to length 16. That's how long it will take to do that under the temperature conditions that I had from my crime scene. Whereas if I only see a maggot that's of length 7.5, then I would, or just be under 8, then this is the most likely temperature profile, growth profile that I would have seen for that particular maggot. And of course what you realise is they don't line up because this is the early temperatures that have been seen in the crime scene. This is the late temperatures, so these temperatures here correspond to these temperatures up here because of course they're experiencing them at different times if they got to different lengths. So what do we see at the crime scene? Well, the particular case that we looked at was a body discovered in late summer in northern England. It had been outside. There was quite a lot of evidence that it hadn't been moved, so it was in a fixed location, so therefore the fluctuations in the temperature were assumed to be related to the outside. There was larva collected and instar assessed. We just used the length of the larva, and it turned out the 8 biggest larvae that Martin discovered were between 14 and 17 millimeters long, and the police required a post-mortem interval estimate. So these are the temperature-dependent. These are our evidence that we collected that Martin did for this particular species of fly, and they're the experimental data. And these are, if you look here, this black line here is the length that you would expect to see for an average maggots of length 14 to 17 millimeters long, and this is a blow up of this region. It says, so it basically suggests that these are the experimental curves, and this black one is what happens when you expose a maggot to the temperature fluctuations that we saw earlier. This shows how the temperature fluctuations actually change the growth. Sometimes it goes faster, the growth. It's always going positive in this case because we haven't reached the wandering stage yet. But here, for example, is a nighttime period where it's actually growing very slowly, whereas here it's a daytime period. It's got much higher temperatures, and therefore it's growing much faster than we would expect of this average temperature. It doesn't do badly for the average temperature, which is why Martin's constant temperature model isn't a particularly bad one, but it's definitely different. It's definitely waving around that particular temperature, and the estimate that we get out is around 150, and the estimated from actually growing the samples afterwards is somewhere between 135 and 178 hours for this. So it was sort of somewhere in the middle, which was reassuring for the modelling. So we're very much a preliminary stage. We're kind of getting this validated in both the multiple species and with the evidence of the sort of known experimental evidence of doing this using the sort of standard accumulated degree hour models. But the way we do this is by proposing a model that's both registration, which is this time shifting, and also the underlying growth curves that you see for particular maggots. And it's useful because we need to predict this unknown post-mortem interval. We need to somehow understand what the post-mortem interval looks like. There are lots of issues that still need to be addressed. There's a lot of theoretical understanding of what's going on in this model. But probably more interesting is the fact that we need to actually look at including instar development stage as well, because this can help us make the model more identifiable and certainly more reduce uncertainty. We've got some preliminary ideas about how to do uncertainty estimation, some Bayesian, some frequentist ideas, but we really do need to understand what the role of the uncertainty in temperature modelling is, what the role of uncertainty in the growth modelling is, and then the uncertainty that we see in the actual estimates of the individual maggots, which have very different lengths, and therefore you want to sort of build that whole uncertainty into the final uncertainty estimate of your post-mortem interval. And then probably somewhat more technically interesting is how you take into account that you may have multiple species. So of course this was one particular blowfly species. Often there are many different blowfly species which have different growth curves. So for example here, this is a simulated experiment which shows two particular growth curves for two different blowfly species, and what would happen if you took estimates from these two blowfly species? Well, in actual fact, what you would see, if you took any individual blowfly species, for example this one, you would get that the model would need to get to this length because that would be the average of these, whereas in this blowfly species it would have to get to the middle here, so it would get a shorter length, whereas in actual fact if you combine the two together, of course you get something that's sort of in the middle between the two, because it's sort of each one of the species is contributing different information. So therefore you need to actually build a model that jointly takes into account the information from multiple species, using the same kind of techniques, but just building it in a slightly different way. And with that, I'd like to thank you very much.
That's moved on along since 15 years ago. I just want to make a point that it's not necessarily post-mortem interval because it's access, fly
it's time to
access. And you
colonisation.
can have an outside situation, but the body's in a vehicle and the flies have got to find it.
Absolutely.
The other thing is, what surprised me, you've got the weather station information, and are you not doing any adjustment to those data, because what you get at the crime scene won't be the same as the weather station.
No, absolutely, no, I completely agree. So in actual fact, this is exactly what we're doing here. Oops, excuse me. Here. So this is the weather station data. This is the temperature logger from the crime scene. We're doing a regression to say how do I calibrate and then do exactly.
The other thing is, any maggot mass is hugely hot.
Yeah.
So if you've got your large maggot from the middle of a maggot mass, they're not subjected to ambient temperature.
No, sure.
Are you actually accounting for that?
So I don't know whether Martin is accounting for that in the way he's doing the experiments, because that would
there is some literature on that.
I think because of if you presume that you set your experimental data collection of the growth curves under constant temperature up in a similar way to how you would expect to see them in the body, and then also then record the lengths of maggots that are inside the maggot mass, then you would some way account for that. But I agree that's an interesting one.
It can be an enormous difference.
No, I'm sure. Yeah.
Okay, there are other questions?
Thanks.
You can ask other questions.
Yeah, that's fine.
I mean, you were first.
A practical question. What about influence of humidity, wind, rainfall? All these things, I suppose, they affect.
Yes.
So
we're not accounting for those in the model. That's absolutely true. But it would be interesting to do. So I believe there are sort of other data sets starting to become available that may have that kind of data that we could actually just build the model on top of. But at the moment, no, it's in a much more controlled experimental environment that the original curves are being collected.
David?
So I've got a really basic question. So what's a blowfly? These things are huge, up to nearly 2 centimeters, right? These are big flies. I think of those as being in the Australian outback bothering sheep.
Oh, I think it depends on the species.
Okay.
So yes, you've got really little ones that are sort of.
Okay. So it is. But is a blowfly include everything that we would normally call a fly?
Yeah.
Or,
Okay. Certainly. Yes. So certainly in this case, yeah.
Okay. So that explains.
So these are not that big.
Because I think of sort of.
Yeah, blue bottle. That's a very good blue bottle.
Green bottles.
Yes. Okay. All right. Next.
Yeah.
It's not.
Yeah, the question which is related to the last one of Patricia about the dependence on, for example, the numerosity of the larvae.
Sure.
So, okay, first because of the temperature, and then also because of the food. Somehow they don't have access to it, and also on the part of the body where you find them.
Sure. Yeah. No, I think I agree that all these need to be.
How does this connect with, let's say, doing some kind of Markov or even dynamic kind of Bayesian modelling of so the effect of temperature, obviously dependent on the previous temperature.
Yeah, so it's very similar. You could kind of see it as a continuous time analogy of that.
Right.
Now, the way we intend to actually one of the ways we've started to look at uncertainty is to incorporate those kind of structures into so that we can put priors on the underlying uncertainty and propagate that through to try and get uncertainty back on the final time to colonisation.
Okay.
So are there any predators of the larvae, like birds or?
Yes, beetles.
Oh, excellent, thank you.
There are lots of beetles that go for them. Staphylinid beetles.
Could that distort, what actually are the original larvae that were on it if they've been eaten?
I'm absolutely sure it could. However, I trust Martin to know what the data he's collecting, has been subjected to, and so I think he provides us with excellent data. I can't fault him for the data he's provided us so far. So I'm sure that all these things are in his mind when he does this kind of.
More questions? I have a question.
Sure.
Did you think of combining it with the estimates from pathologists? So they will temperature the body and.
So I think at early times that's a very good, and I think Martin provides one set of evidence and the pathologists provide another and they combine together. Whereas I think for longer durations, I'm not sure how useful that is anymore.
But they still have estimates that should be combined at some point.
Yeah,
I think.
absolutely. And I think that in some sense, using all that is the sort of how you then quantify the uncertainty at the end. You want to be able to use all that evidence to try and get a true uncertainty on the post-mortem interval, accepting that there are many uncertainties, such as the colonisation time, such as the uncertainty in the inherent pathological evidence, for example.
Could I just ask how many temperature readings are taken at the crime scene a day? Because you can take them every hour.
Yeah. So I mean, you mean in the logger?
Yes.
Yeah, we're using hourly time.
Oh.
Hourly. I mean, I think you can get it on a sort of minute-by-minute basis.
Yes,
It sits
yeah,
there, but we're using it as an hourly measurement.
If there are no more questions, then I'd like to thank you very much.
Thank you very much.